Welcome to Clinical Histology. I'm Dr. Xie from SUMC. In this session, we will focus on the nephron, the filtering unit of the kidney. Firstly, we will take a closer look at the kidneys and identify their nephrons. Then, we will explore the components and basic functions of the nephron. We will also examine the layers of the filtration membrane, a vital structure for filtering blood to produce urine. The kidneys are two bean-shaped fist-sized organs located in the retroperitoneal space, one on each side of the spine. They, together with the ureter, bladder, and the urethra, constitute the urinary system. Healthy kidneys filter about 90 mL, that is a half cup of blood, every minute, removing waste and actual waters to make urine. As shown on the right, the hemisected view of the kidney reveals three distinct regions: the outmost renal cortex, the middle renal medulla, and the inner renal pelvis. The renal cortex and the renal medulla make up the urine-forming portion of the kidney. The cone-shaped structures in renal medulla are called renal pyramids, which are made of parallel bundles of small tubes. We will see what these tubes are in a moment. The renal pelvis drains urine made by the cortex and the medulla into the ureter. Please note that renal cortex houses 90 to 95% of kidney's blood vessels, while fewer blood vessels are found within the renal medulla. How is the urine made? As the urine-forming portion of the kidney, the renal cortex and the medulla contain over 1 million microscopic filtering units called nephrons. Each nephron is capable of filtering blood and producing urine. This diagram shows a single nephron, which is composed of two major components: the globe-shaped renal corpuscle and the long meandering tube of epithelium, called the renal tubule. The renal tubule extends from the corpuscle and drains into the collecting duct. The corpuscle filters blood to form primary urine. The primary urine then flows through the renal tubule. In the renal tubule, almost all water, along with electrolytes and nutrients in the primary urine, are reabsorbed and returned to the blood circulation. The remaining fluid and waste become secondary urine, which flows out of the nephron tubule into the collecting duct. Please note the arrangement of these components in the kidney. The renal corpuscle and the majority of the renal tubule reside in the cortex, whereas parts of the renal tubule dip into the medulla and form parallel tube bundles with the collecting ducts. Next, we will zoom in to see more structural details of the renal corpuscle and the tubule, which will help us understand how exactly urine is formed. Each renal corpuscle consists of two parts, the glomerulus and the Bowman's capsule. The glomerulus is actually a tuft of looping fenestrated capillaries that arise from the afferent glomerular arteria, whereas the Bowman's capsule is in fact the dilated pouch-like proximal end of the renal tubule. The glomerulus invaginates into the Bowman's capsule to form the renal corpuscle. Therefore, the Bowman's capsule has two layers. The parietal layer is composed of squamous epithelial cells, while the visceral layer consists of modified epithelial cells called podocytes, which are in intimate contact with the glomerulus. This scanning electron micrograph shows a glomerulus surrounded by the podocytes. We can see the podocytes have long food processes, which is called the pedicels. The pedicels wrap around glomerular capillaries and interdigitate with each other, leaving slits between them. The slits are covered by slit diaphragms, which restrict the passage of large macromolecules and ensure that they remain in the bloodstream. These wheeled podocytes are indispensable components of the filtration membrane for blood filtering. The filtration membrane is made up of 3 layers: the fenestrated glomerular capillary endothelium, the podocytes, as we discussed just now, and the basement membrane between them. The filtration membrane is actually a barrier between the lumen of glomerulus capillary and the lumen of Bowman's capsule, so-called Bowman space. Please pause the video and try to identify where the filtration membrane is in this diagram. Part of the filtration membrane are indicated by the maroon rectangles in this diagram. I hope all of you can find them. As the blood flows through the glomerulus capillaries, it is filtered by the filtration membrane. Consequently, the filtrate enters Bowman space, which is continuous with the beginning of the renal tubule. As mentioned above, the renal tubule is a winding tube responsible for further processing of the primary urine. It has 3 regions: the proximal tubule, constituted by convoluted and straight sections, the nephron loop, and the distal tubule, constituted by convoluted and straight sections as well. They differ in their structures and functions. As shown in this figure, the proximal tubule consists of simple cuboidal epithelial cells with central nuclei and prominent microvilli that project into the lumen to form the brush border. This border greatly increases the surface area for efficient reabsorption. A great deal of secretion takes place in the proximal tubule as well. The proximal tubule continues as the thin limb of the nephron loop, also known as the loop of Henle, which is hairpin-like structure that extends deep into the medulla. The nephron loop has 2 limbs. The descending limb travels down to the apex of the renal medulla, while the ascending limb climbs up towards the renal cortex. The descending limb is composed of simple squamous epithelium. The majority of the ascending limb is made up of thicker simple cuboidal epithelium. The final segment is the distal tubule, like its proximal counterparts. The distal tubule consists of simple cuboidal epithelium. However, the distal tubule lacks a brush border. For more detailed functions of the renal tubules, please check your textbooks or our online clinical physiology sessions. Glomerular nephritis refers to inflammation of the glomeruli in the kidney. It is characterized by the leakness of the filtration membrane, which will lead to proteinuria and/or hematuria, and finally complete renal failure. Biopsies of the kidney is an important way to diagnose the different types of glomerular nephritis. The left shows a normal glomerulus surrounded by Bowman's capsule. The Bowman space can be easily identified. However, in the section on the right, from a glomerular nephritis patient, the proliferation of fibrous tissue has occupied the entire Bowman space, and it is difficult to clearly identify the glomerulus. It seems that the glomerulus has been compressed and replaced by the fibrous tissue. These abnormalities will destroy the normal functions of the nephron and cause renal failure. Almost all the major functions of the kidney are performed by the nephrons. In this session, we have examined the structures and basic functions of the nephron. The nephron is composed of a renal corpuscle and a tubule. In the corpuscle, the pressure in the glomerular capillaries forces water and small solutions of blood plasma to go through the filtration membrane into the Bowman's capsule. From the Bowman's capsule, primary urine enters the long renal tubule for further processing. The processed filtrate will finally flow out of the nephron and drain into the collecting duct. Can you describe the components of the nephron and the layers of the filtration membrane now? That's all for today. Thank you for joining us. Hope to see you soon.